Hi all, my name is Griffin and I'm going to be talking to you about how insulin action models applied to glycerol and glucose yield different dynamics. Now first, I'd like to thank the authors and the organizations that made this work possible up front, so big thank you to them. Now, before we get into talking about how the kinetics of glycerol and glucose are different, first we want to talk about some of the physiology behind these systems. Now, when we're talking about glycerol and glucose, we're talking about metabolism. And one of the main goals of metabolism is to distribute energy across the body to places it needs to be. Now, in the fasted state, we're relying on our energy reserves or our endogenous sources of energy to be able to distribute this energy throughout the body. And for example, you have tissue like your liver, hepatic tissue, that will release glucose so that it can be distributed throughout the body. At the same time, your fat tissue or your adipose tissue will be breaking down triacylglycerides by lipolysis to produce glycerol and fatty acids. These fatty acids are taken up by your muscle tissue to act as a source of energy. Now, we're not always in just the fasted state, but we also eat food. We take in food, and this will increase the blood sugar concentration, or our glucose concentration in our blood. And then this glucose that's readily available, it needs to be taken up by your tissues so it can be either stored or used as a source of energy. Now, at the same time that we're using this exogenous source of energy, we no longer need to rely on our energy reserves. So we can decrease the lipolysis that produces glycerol and fatty acids. As you can see, the arrows that produce glycerol and fatty acids have shrunk in the fed state. But how are all these pathways managed? And it all comes back down to insulin. So insulin regulates glucose and glycerol dynamics by binding to insulin receptors on the cell and then signaling metabolic changes. This influences glucose or increases glucose uptake and then also suppresses lipolysis to decrease glycerol concentrations. Now, when more insulin than normal is required to achieve that same metabolic control, this results in a phenomenon known as insulin resistance. And insulin resistance can be tissue specific. For example, it might require more insulin than normal to suppress lipolysis sufficiently, making your adipose tissue insulin resistant. Now, insulin resistance is an important measure to be able to observe early, as it is a marker on the progression towards metabolic diseases like type 2 diabetes. So we know it's important to capture insulin resistance, but how do we observe insulin resistance? And this is done through techniques like the hyperinsulinic euglycemic clamp. Now, this is a medical technique that deals with steady state values where glucose and insulin are infused into a patient in order to suppress specific tissues. Now, different tissues are suppressed at different insulin concentrations, and this establishes a dose dependence associated with the tissues and insulin concentration. We've observed that adipose tissue and lipolysis are suppressed at much lower levels of insulin than glucose is encouraged for uptake. Now, this is a static technique and it's non-physiological. So we're interested in looking at the dynamic protocol because we are dynamic systems. And this comes from using tests like the oral glucose tolerance test and systems of ODEs. We're going to be talking about that side. Now, using these systems of ODEs and tests like the oral glucose tolerance test, we're going to be aiming to determine if the kinetics of interaction between glucose and insulin are significantly different than those of glycerol and insulin. So how do we get this data to represent these metabolic pathways? It all comes from an oral glucose tolerance test. So in an oral glucose tolerance test, the patients undergo an overnight fast and then are given a sugary drink, a glucola drink. Now, one of our 10 participants that we looked at can be seen here taking that glucola drink. Now, over the protocol duration, their blood is sampled at specific time points. And from these blood samples, metabolite concentrations of glucose and glycerol are extracted, as well as concentrations of insulin. Now, you can see in response to the drink, there's an increase in glucose followed by a decrease of glucose taken up by the tissue. We also see that glycerol decreases towards that suppressed state and returns back to baseline. And then we also have insulin increasing to act on these tissues and then decreasing. Now, in a typical glycemic or a participant of typical glycemic control, insulin would only need to go up to about 200 units. In this case, we can see we're going well above that. And this is a marker of insulin resistance. So this population that we were looking at is an insulin resistant population. To measure glucose dynamics, we use the oral minimal model that was developed by the Cabelli group. In the oral minimal model, there are two sources of glucose. the drink, the glucola drink, and then the endogenous glucose production that comes from the liver. Now, insulin acts on glucose by influencing the uptake. And to determine insulin action, we're feeding insulin concentration into a compartment known as insulin action. And insulin action isn't exactly insulin, but it encompasses all of the signaling that results in glucose uptake. Now, the model that's related to the schematic is here. And you can see, and this term is our drink term, our drink input. We have our endogenous source of glucose, and then we have our uptake term that is a function of insulin action. Right here is X. Insulin action is X. And insulin action here changes in response to the insulin concentration above baseline. So this is insulin influencing action on the tissue by creating signaling or by influencing the signaling of this tissue. To capture glycerol dynamics, we developed a model. In this model, we have two sources of glycerol. The insulin-independent lipolysis is the source of glycerol that is unaffected by insulin. Right? As we saw in our original schematic, some glycerol is still produced in the fed state. This accounts for that. The second term is insulin acting on lipolysis, suppressing lipolysis, so not as much glycerol is produced. As you can see, insulin action is suppressing lipolysis. Now, this insulin action is unique from insulin action on glucose because they are two different systems, but is a similar principle in the idea that insulin is fed into a signaling pathway that then influences lipolysis. Now, here's our model associated with that schematic. You can see that we have an insulin action-dependent lipolysis function, L of X, right here, that when we expand it, we have a constant term for our independent insulin term, and then we have an insulin-dependent term depending on insulin action, X here. Again, insulin action is a function of insulin above baseline, influencing that signaling. Now, this is the data and our fits to the data. In both figures, you can see that we are feeding in the same insulin concentration. So in both figures, you have this red as insulin. Now, the blue is the metabolite. So on the left, we have the metabolite of glucose, and on the right, we have the metabolite of glycerol, and that's the data. Now, the black is the numerical solution that we have from fitting the data with our model. Now, you can see that we're getting good fits, and everything is lining up nicely. But each of these models also come with insulin action. And you can see we have glucose on the left again and glycerol on the right. Now, off an initial just observation, you can see that these are different curves. And we're going to dig into some of the features that make them different. Now, there are 6 measures that we use to evaluate differences between these two different insulin action curves. The two most important, bolded in the table on the left, are peak timing and normalized end offset. They convey differences in the dynamics. So the peak timing is the time after the drink that insulin acts greatest on that tissue or on that metabolite. The normalized end offset is how far back from baseline that action is at the end of the protocol duration. Now, what we can see is that glucose is consistently larger in terms of peak timing, meaning that insulin is acting on glucose tissue after it's acting on glycerol tissue. We also see that the normalized end offset is greater for glucose than glycerol. In this case, insulin is acting on glucose tissue much after insulin has been acting on glycerol tissue. We weren't just interested in measures, but also in how the metabolite and the insulin action were changing relative to each other. To do this, we used phase plots. And in a phase plot, we have our insulin action normalized on the abscissa and our normalized metabolite on the ordinate. Now, you can see our glucose is in black, and we have a cyclic pattern here where our metabolite will increase, our glucose will increase, while insulin stays relatively constant. Insulin will increase while glucose stays relatively constant. And then the opposite, in a you-go, I-go fashion. In contrast, glycerol is going to decrease while insulin is increasing and then going back, right? So we've got a crescent shape here. So let's unpack these phase plots a little bit more relative to the time series they come from. So we can see on the far left, we've got our glucose time series and our phase plot for glucose. As we saw, it's a cyclic pattern, a you-go, I-go pattern, right? Now, if we were to see the same type of shape in glycerol, or our hypothesized glucose-like glycerol, we would expect insulin action to increase, followed by a decrease in glycerol as insulin action stayed constant. Of course, we don't see this cyclic pattern of you go, I go for glycerol, but instead see this crescent-shaped pattern. And this crescent-shaped pattern implies that insulin action and the metabolite are changing at about the same time. So you can see here, the slopes cross each other as opposed to crossing near these flat regions. Now, this indicates that glycerol and glucose are kinetically different. They don't have the same kind of relationship with insulin that the other does. So all of these observations from the modeling suggest that the kinetics of insulin action on glucose differ from insulin action on glycerol. Specifically, glycerol changes more quickly with changes in insulin concentration. And since glycerol is associated with adipose tissue, this indicates that adipose tissue responds more quickly to insulin, while muscle and liver tissue have a delayed insulin response. So these are preliminary results observed in a population that is characterized by high insulin resistance. And we're interested to see if these same observations would persist in a population with more typical glycemic control. We're also interested to extend these observations to a larger population of our own. So I would like to thank my mentors, as well as all the funding I received, and of course the participants and their families that contributed to the study and made it possible. Thank you all for listening, and I look forward to answering any questions.